So let's say you've identified a module of interest using Tool 1 or Tool 2. You then used Tool 3 to examine annotations enriched in this module and identified experiments that similarly activated, which can be very informative. But of course, modules contain many genes, and it's unlikely that every single module gene, even for a highly active module, actually changes in your experiment. So you may want to know which specific genes in the module of interest change the most in your experiment, and are they the same genes that change the most in another interesting experiment that you've identified using the module annotation page in Tool 3. For that, we have created Tool 4. So let's take a look at it now. Here, you can compare gene expression between your dataset and any of the 716 experiments from our compendium. Or you can compare two different experiments from the compendium to each other. First, you select which you want to do. Let's say I'm interested in module 66 because by using Tool 1, I found that it's the most highly active module in my ISP1 gene expression data. I then examined the annotation page for module 66 and saw that this module is highly activated by paraquat treatment and also interestingly by comparison of L4 to L3 larvae. So here I select two experiments from the compendium and I copy and paste the experiment IDs for these two experiments into the corresponding fields. So this will be experiment 1 and this will be experiment 2. I think these are entered as defaults, but I'm just showing you where the experiment IDs go. So then you select the module you would like to highlight and press submit. I get a plot that shows gene fold changes in experiment 1 on x-axis plotted versus gene fold changes in experiment 2 on the y-axis. We see that the overall correlation between gene expression changes in these two experiments is 0.57, while the correlation between genes that belong to module 66 is even better at 0.7. I also see that the vast majority of genes in this module belong to the A hemimodule, which is not surprising because by design, hemimodule A is the dominant hemimodule, meaning it contains more genes. I can mouse over the genes and get their names, like so. For example, I see that these genes change the most in response to both paraquat and in L4s relative to L3s. It's very important to keep track of the direction of the contrast. The first experiment I put in, if we look here, compares untreated animals to paraquat treated animals. And the second experiment compares L4 to L3 larvae. So even though the gene changes are positively correlated on this plot, they are negatively correlated if we're talking about the comparison of paraquat treated versus untreated to L4 versus L3. That is to say, genes in module 66 decrease in response to paraquat treatment and increase in L4s relative to L3s. So just imagine flipping the x-axis. You can download the results table here and manipulate it to your liking. The results are also displayed in an interactive table below, which you can sort in various ways and search for genes of interest. I can also compare my own data set to a data set from the compendium. For that, I choose this button, and let's use the ISP1 mutant data set again, the same one I used for Tool 1, and the one that's provided as a sample file for you to try. So we can download the sample file. This is what it looks like here. So you too can use the same file you uploaded to tool 1, so long as it contains 2 columns, one with worm base IDs and the second with fold changes or log fold changes for every detected gene. So I downloaded this table. Now I'm going to upload it here. Whoops. There we go. So that will now be experiment 1. And for experiment 2, let's say, again, I want to compare to L4 versus L3. So it's already here, but here it is. And that's it. So I highlight the module I want to focus on. I press submit. And I get the same sort of outputs as before. So by doing these 2 comparisons, I learned that almost all genes in module 66 are downregulated in my ISP1 mutant experiments. They're also downregulated by paraquat treatment and upregulated between L4s and L3s. I could now examine the plots in the table to identify genes that change the most for follow-up analyses.